Good afternoon, everyone. Greetings from Melbourne, Australia. Thank you very much to the organizers of DigiCrop for inviting me to speak to you today. It's too bad, of course, that we all couldn't see each other face to face. However, hopefully by doing this meeting digitally, we will be able to communicate and interact with far more people than we would have. Right, so my area of focus for my research throughout my career has been all on plant roots and their interaction with the soil. And I really feel that the technologies that are now at the fore and also receiving a lot of attention for improvement from sciences outside of biology really can be turned to, in part, allowing researchers and practitioners to quantify roots, soil, interactions with soil in this whole space called the rhizosphere to help us realize some of the potential from improving the below ground processes for agriculture and the environment for saving land, water, and also fertilizer in this 21st century. Now, in my definition of the rhizosphere, I include the soil space that has both the plant roots and all its associated symbiotic organisms. There was, I guess, an ongoing phase where roots were actually separated from the soil in this definition of the rhizosphere. So the rhizosphere had a definition of being the soil outside of the root influenced by the presence of the root. But it's much more conceptually useful to combine both the root and the associated soil that is influenced by the root in one zone termed the rhizosphere. And this is the way I'm going to refer to the rhizosphere in this talk today. Now, just by way of review, here is a plant root system. And it has been dug out of the soil. And this is a wheat plant. You can see the beginning of the shoots here. And this is its root systems at about 4 or 5 leaf stage. There are many different types of roots that make up this root system. And a lot of the roots are binding soil very tightly. If we put one of these root tips where the root is growing under the microscope and stained and put a special dye to be able to see any microorganisms associated with the root, this is what we would see right here. This is a tip that is covered in what's called the root cap, these very special big root cap cells. And all of these pink and blue dots are bacteria that are associated with the root. And this is really the genesis of the rhizosphere. A root tip arrives in a volume of soil. It exudes different compounds, largely sugars, carbon-containing compounds. And these are used as food by microorganisms. And each millimeter of root length on a plant has roughly about 1 million microorganisms associated with it. Now, this image here shows the underside of the wheat crop that has not been washed or disturbed. And many roots of different diameters, this is a wide diameter root, this is a fine diameter root, medium diameter root, are occupying this soil volume. And this gives a sense of the density of roots that are below ground under almost all plants. And so the rhizosphere as I will use the term, is the fear of soil that is influenced by roots, and it includes the roots and their symbiotic organisms. The rhizosphere is a very complex spatial and temporal zone because it has root systems, parts of roots, and microorganisms of different ages, different functions, and locations at any point in time in a column of soil. And this diagram here illustrates the dynamics of this space and time, let's say age dimension of the rhizosphere. Starting again, looking at a root tip. This is a diagram of a root tip. These are root cap cells, shown nicely in the micrograph in the previous slide. And as I said, when a root tip arrives in a volume of soil, this is the genesis of the rhizosphere, and in fact the genesis of deposition of plant organic matter into a volume of soil. Over a period of time, 4 or 5 days or so, this tip tissue, this biological tissue, differentiates into more mature root cells, so that in this same volume here, it is now occupied by newer cells that have formed into root hairs, projection out into the soil, and underlying within the tissue cells that function largely for the transport of nutrients and water taken up from the soil, but also transport of sugars and amino acids and other compounds coming down from the shoot into this root tissue to allow it to grow and function. A week or two later, this same tissue here, excuse me, differentiates, continues to develop, and it will make a baby root, a branch root, and it has its own root tip and root cap cells. Sometime after that, this same piece of root, excuse me, differentiates into an older root again. The cortex, this outer region, may senesce and slough, so providing even more complex and different compounds for microorganisms. And a branch root, such as this one, could develop its own branch root. All of these temporal events essentially overlay each other. And so in every volume of soil, there has been a succession of root cells of young through to old ages, and a succession of microorganism communities that have been feeding off of and transforming the organic compounds in such a zone of soil. And I've just taken all of these diagrams here and overlaid them on top of each other to give you a sense of the complexity and the, I guess, evolution of biological material once a root arrives in a patch of soil and progresses through its biochemistry and interactions with soil microorganisms. And this is really the spatial and temporal complexity of what it is in the rhizosphere, the root systems of every plant that we see outside, in natural systems and in managed agricultural systems. So really the biggest problem for agriculture and environmental sciences has been and continues to be the quantification of this rhizosphere biomass. And this is another view again of the organic material that we can see with our own eyes in the rhizosphere, which is really the root biomass material, but this itself is associated with microorganisms, which have their own biomass, and also efflux and exchange of elements between the soil and the root and the microorganisms. This image here was generated by allowing these maize roots, this is a maize crop here, to grow up against a clear face, allowing all the roots to become pressed into 2 dimensions, and a photograph can be taken to indicate the quantity and the root length and the directions of root systems under a crop. And the challenge of quantifying this rhizosphere biomass has been with researchers for over 100 years, since the beginning of crop improvement efforts using both management and breeding, which largely commenced around the beginning of the 20th century. Today, crop growth models and yield gap estimates have a low or no estimate for rhizosphere biomass. And third, there are new and existing technologies that we really need to make use of to quantify the rhizosphere biomass in order to keep making improvements for agriculture and the environment in the 21st century. And I'm going to talk about these 3 points today in my talk. So this first point, the challenge for researchers for over 100 years has been quantifying these below ground allocations of biomass from the shoots of plants, and in turn, the return of water and nutrients up to leaves from that root biomass. And this is a grass plant. This is the shoot. This is its root system, been washed from soil. And I'm just illustrating with these arrows and these question marks to indicate that the carbon and nutrients that go down to the rhizosphere are quite poorly estimated. Very wide range of values have come from experiments to put a number and an estimate on this below ground allocation of carbon that has been fixed in the leaves and converted into different biochemical compounds, and the extent to which sugars, amino acids, and other compounds go and are allocated below ground. And in turn, approximately a few hundred water molecules are required for each carbon that's actually fixed in photosynthesis. And the water and the nutrients, particularly the nitrogen to build rubisco in the leaves, moves from roots up into the shoots. And we have quite a poor understanding of the quantification of these 2 fluxes. And it's interesting to have a bit of a look at the history of where today's concepts in crop improvement come from. And really, they avoid largely the need to directly measure rhizosphere biomass. Here's a timeline from the beginning of the 20th century through into the beginning of the 21st century through to today. And what I really want to highlight to you here is that there have been a few key trends in, I guess you would call it plant physiology and crop sciences, to allow researchers and breeders and agronomists and modelers to make estimates about the biomass produced by plants, the resource efficiency of that biomass, and the amount of that biomass that goes into the grain, the harvested product. And it really started critically around 1913 in the US. Briggs and Chance, they introduced the water requirement concept, and they watered plants in pots, and they measured shoot biomass, as far as I can tell, possibly root biomass, but the water that was, and they weighed the water that went in to allow the plants to grow. And this was really the first introduction of a concept that biomass of plants requires a certain amount of water. And sometime after that, in the Netherlands, de Wit, a key sort of original researcher underlying some of today's crop models that come from the Netherlands, he got an empirical, so a measurement-based relationship between yield and transpiration. And so this was a continued building of the idea that the plant biomass and then furthermore yield requires a certain amount of water. Now, a little bit after that was introduced the phenomenal description of leaf photosynthesis and its trade-off with transpiration. This was really pioneered by Slatcher and co-workers, and it's the foundation of today's Farquhar von Cameron photosynthesis model, leaf-based model in many crop growth models, as well as environmental climate change global biomass models. And this was termed the water use efficiency concept, and it's based on photosynthesis. So an input of carbon with an exchange of water based on a leaf. Subsequent to really this phenomenological and very important concept of looking at plants from the point of view of leaves, there was the introduction of the importance of radiation. And Schibels and Weber in 1966, I think in the UK, they made empirical measurements of plant biomass against the radiation intercepted. And again, I was not able to tell whether they included roots in the biomass. I think it's just shoots, but it really began the idea that plant growth could be predicted from the radiation that is intercepted by leaves. And this was the Monteith concept, 1977. Crop biomass is a basis of radiation, can be based on the radiation intercepted. And really, after this, in 1986, Sinclair in the US, really the pioneer behind the main crop growth models that come from something called Cirrus. He really took these above ground concepts, so carbon input into the plant and biomass can be estimated from the radiation that's intercepted. So the sun and the leaf area index. He added on the importance of air temperature for estimating the biomass from solar radiation. And independently or separately, Ritchie, again working on water, introduced the water limiting concept. And this is really to point out that crops in drying soil, their physiological responses, growth, photosynthesis, can be characterized as a fraction of the soil water that is available and remaining for growth. Sinclair's model critically, however, needed to estimate the amount of the biomass generated in the leaves that went below ground. And he used some actual data from Bray in 1968, who weighed root systems. in pots and also in the field. And Bray had a range of numbers, and the mean was that the dry matter accumulation by roots was 25% of the total dry matter accumulated in the complete plant. And so today, crop growth models, many of them coming from Cirrus, estimate below ground biomass as 25% allocation of the total plant biomass, with a function related to canopy, radiation, the photosynthesis model coming from Farquhar and Slatcher, soil water extraction related to the Ritchie concept, and air temperature. Now the question is, not really a question, but I think it's timely to look again at whether this number of 25% that was obtained from data in the 1960s should be revised or re-examined, remeasured, etc. And taking some numbers from 3 studies here, independent numbers of different plant sizes and genotypes, but all of wheat. The percent of total biomass that's below ground biomass from these studies ranged from 50%, 69%, down to 3%, with a mean of 41%. Different stages, both field and controlled environment experiments. This mean would suggest that current models are underestimating the below ground allocation, but also that there's a very, very large range that may depend on plant age, as others have pointed out, may depend on species, and may depend on growing environment. Now, I'm going to look now at yield gap estimates. Yield gap estimates are very important for advising research, farmers, and practitioners as to whether yield gains can be made through the increased grabbing of soil resources, the increased production of biomass, and the increased production of grain, or harvest index, the allocation of biomass into grain. And these components have been pulled together in what's known as a yield identity. So yield is a function of the particular resource that's used by the crop, the biomass that's generated by the crop, times the harvest index, the allocation of the biomass to the grain. Now, this yield identity has been around for a long time, first originally in the horticulture sciences. Then in the crop sciences, it was really brought in by Donald and Hamblin, and they don't actually specify whether biomass includes root biomass. The issue is, however, that today in yield gap analysis, root biomass measures are not included. And this is some reviewing of the literature and conceptual, I guess, sort of analysis that I've been doing with Gupta Vedakatu at CSIRO recently, where we are digging into the crop growth models. I mentioned the history earlier, to see where values and concepts have come from, and if there is a gap in the below ground concepts, and I'm suggesting that there is. But also, we've been looking at yield gap analyses. So the way yield gaps are identified are by plotting the yield of different crops, or the same crop, or different varieties, different yields that are obtained against the resource that is supplied for those yields. And the resource can be things like soil, water, nutrients or fertilizer supply, the amount of radiation, even land area. And the result is a range of points on such a graph. So for a given amount of yield, a given amount of water, for example, the crop can yield this much, or it may yield this much. Less resource, it may yield less. More resource, it may yield more, with a potential boundary line that, depending on the crop, this might be, like, for example, for wheat, 22 kilograms per hectare per millimeter of water. And if growers and a certain land area is not reaching this boundary, then there is a gap in achieving that yield, not related to the genetics of the crop, but needing more resource. But it's also possible to raise the boundary of this line to a new potential. And this can be done with modified genetics of the crop, modified processes within the crop, physiology and function, or it can be done by increasing the amount of resource that's acquired by the crop. And what does this mean? Well, since below ground biomass is not included generally in yield gap analyses, then there are opportunities for more yield and resource efficiency if it were to be included. This is what could happen if the below ground biomass was included. There is opportunity, therefore, for more yield through greater harvest index that may draw carbon resources in allocation from the rhizosphere into the grain. There is an opportunity for more yield through greater above ground biomass with roots that use less carbon for their functions, some more efficient root systems. And finally, there is greater yield through more resource capture by root systems with better functioning roots in terms of their spatial colonization, possibly their longevity, and deeper rhizospheres. So finally, in the last part of my talk, I'm going to talk about the technologies that we have today and what we need to put below ground quantification into our crop improvement research and applications generally globally around the world. So first what I'll point out is that it's very important that, in my view, that we propose concepts that are based on dynamics, space, time, and the forces that are underlying the changes in roots, microorganisms, and elemental flows, carbon, water, and nutrients in the rhizosphere, because these are changing over time. And in this paper that I wrote with Boriana Arsova and others, we conducted modeling, and we showed that the way roots change their allocation of carbon, particularly between thicker primary roots and branch roots, thinner roots with fewer cells, energy, the energetics of transport, changes. And so the amount of energy used by roots coming from photosynthesis changes. And by plotting shoots and roots in a space and time axis, it becomes it becomes feasible to conceptualize the dynamics that could be important for integrating and applying below ground changes. Now, underlying this macro view of root systems and macro view of their anatomy, each cell in the root has different sorts of transporters for the uptake of water and nutrients, but also for the flow of carbon from the shoot to the root. And Borriana mapped from the literature the different transporters that are known for different species in roots along these root ages that I pointed out in the initial slide. Remember, I mentioned that roots go age, they differentiate. And with that differentiation, they change their transporters. And this means that the dynamics underlying these changes are very important to initially recognize in case they should be embedded in a much more nuanced and flexible allocation pattern in crop growth models and vegetation estimates. Today's option to measure the rhizosphere, remember I'm always including roots in the rhizosphere, are non-invasive and minimally invasive. Actually, in this recent review in Trends in Plant Science, we showed all the technologies that have been reported in the literature. In controlled environments, some of them are very, very early in stages of development. Some of them have really shown to have very limited uses. And some of them are very exclusive to certain laboratories because of their expense. And some of them can only be used in controlled environments. Very few of them can be used in the field. And researchers have really used the entire spectrum of electromagnetic radiation to find light waves that can actually penetrate or semi-penetrate in soil or on the faces of soil to quantify roots. I don't think that we have harvested and harnessed the data that has come from the studies from all of these instrumentations. We can do better than the 1960s value of Bray. In the field, we're still limited to either coring or shoveling, or the insertion of windows, mini rhizotrons for imaging roots, or non-invasive sampling methods, canopy measurements, and also EM38, so salt and water measurements, but from above ground. And in my opinion, and also that of PhenoRob is a very exciting investment by the German government and combines Jülich Forschungszentrum and the University of Bonn to really put everyone's heads together to quantify crops more accurately. And I would suggest that the below ground measurements is really possibly the most critical area for advancement. And that advancement can probably be made by using a hybrid of both shoot and root measurements, field and controlled environment measurements, and dynamic concepts. One of the exciting thing is that the phenotyping technologies that have been developed largely by Uli Schur and his institute at Jülich, the sensors, the concepts behind the sensors are very, very similar or can be adapted to the sensors that are used on fields. And this makes the lab to field hybrid possibility a reality. So with that, I just want to thank my colleagues, past and present. Really want to thank my colleagues from CSIRO. I was based there between 2001, 2015, greatly influenced by people like Richard Richards and John Passura. And then I'd like to thank my colleagues at Jülich, the Root Dynamics Group, which is now led by Boriana Arsava and Johannes Posma, who you all know. And now I have joined the Melbourne plant scientists, and very excited to be here. And I guess in terms of our own research, we will be taking a closer look at the fundamental dynamics, the kinetics and the forces of carbon movement below ground. Thank you.